Hello everyone and welcome to Let's Crack NEET UG English, and we are on the chapter Respiration in Plants, and we are talking about aerobic respiration and its steps, and we'll continue with the same. So I guess people will start joining in. And we plan to complete electron transport chain and oxidative phosphorylation. These are the last two steps, which we will be discussing today. Yes, Hiren, good afternoon. How are you? Did you finish the homework which I gave you, the questions? That's really great. In the previous class also, people joined like couple of minutes late because they thought, or rather they had some other class and then they had to switch from that class to this one. Yes, I was just typing. Okay. Okay, so like in couple of minutes when everyone joins, then we'll go over those questions and then we will take up electron transport chain and oxidative phosphorylation. Where are you based, Hiren? Where are you from? I guess this timing is, oh, you're from New Delhi. I guess this time is a little, it is clashing with other classes, and that's why yesterday also I found that the students came in like 4 or 5 minutes late. Okay, Rashmi, good afternoon. Rashmi, did you get a chance to answer those questions which I gave as homework yesterday? And was there any doubt, anything which was not clear while answering those questions? Okay, you answered, very good. And all of the answers were done? Homework, we get, okay, so Hiren has typed the complete answer. What we'll do is we'll see those questions and then we'll take your answers also one by one so that we know whether it is all or whether these answers are correct or not. Okay, so yeah, Kalai, hi. See, this is what I was telling you, that students are joining in little late because yesterday only somebody told me that they were attending a physics class and then after that they had to come here, so I guess there is an overlap of few minutes. Okay, so what we can do is we can go over the basics, the things which we talk about almost every day, and then we will come to our topic. See, we plan to cover electron transport chain and oxidative phosphorylation in today's lecture. These are the last two steps of aerobic respiration. We did glycolysis, the link reaction and the Krebs cycle, okay? This is about the platform which we are using, and I'm your biology teacher. And you know, this is the app which you need to install, because this is the thing where you would get all those plus programs and iconic programs. I was talking to somebody and I was told that on the plus program there are mock tests which are conducted, and you can take those mock tests. So if you're studying from me here on YouTube channel or through special classes, on the app they are conducting mock tests on the plus programs. There are teachers who are already taking some kind of modules. So you start taking those mock tests, but for that you will have to install the app and take the subscription, the plus subscription, so that you get to know those mock tests and your ranking amongst the students who have taken that test. This is the plus subscription benefit: daily live classes, structured courses, there are quizzes, unlimited access, and this is the newly introduced thing, that is iconic subscription. Here there is one-on-one guidance from one top expert as your personalized coach for your preparation. See, you know, even I'm planning to have a small video for you on 31st at 2 o'clock, and the channel is going to be Biology 101, and that is the strategy to score 360 in biology. So you can join that video, watch that video live with me, and I'll tell you there are certain strategies which you, if you use, you will be able to score 360 on 360 in bio. These are the few differences between the plus and the iconic programs. In plus and iconic, the things which are common are the best educators, the interactive classes, course and PDFs and quizzes. This is common. But in iconic, I was talking about this personal coach, the planner, the guidelines which are given by the experts, and then there is test analysis. And this is the subscription plan for the plus program. So for plus, these are the different options that are available: a month, 3, 6, 12 and 24. And these prices are after discount, like if you take a long-term one. But if you use this code of mine to take the subscription, you will get 10% discount on the subscription fee. This is for plus. And this one is for iconic. So for iconic, you have 3 options. You can go for a 6 monthly plan, a 12 or a 24 monthly plan. Again, here also you will be eligible to get this 10% discount. Okay, so Hiren says I will. You know, all of you can join, because these strategies are very important when you're preparing for such an all India level competition. There are 16 lakh students who are appearing for NEET, and in that, to get a good rank and then get a good college is the most important thing. We work very hard, but there are certain things which are missed out here and there, and this is what we tell you to work on whenever we have such kind of strategies videos, right? So we will be talking about biology and how you can score maximum. Alright, so this is about the fees and the benefits of plus, which I have already told you. Now this is the question, or these are the questions rather, which I gave you as homework, and Hiren has given the answer. Who else has done? The first question is, if you have one molecule of pyruvic acid, how many ATPs will we get on its complete oxidation? In this case, Hiren has given the answer as 12 ATP. Do you have any other answer? Anybody else who has done this? And do you think this answer is correct? Kalai says 1 point. Oh, first question, 12 ATPs. Same as Hiren. Are you sure whether this answer is correct or not? Because we will be talking about this question right now. First question, 12 ATP. See, Rashmi also says 12 ATP. Now, see what is the answer going to be. Glucose broken down into pyruvic acids, 2 pyruvic acid, okay? We are talking about only 1 pyruvic acid. Can pyruvic acid itself participate in Krebs cycle? No. So this has to change into acetyl coenzyme A, and here we get 1 NADH2. Remember, this is the link reaction, and then this participates in Krebs cycle, and Krebs cycle gives us 12 ATPs. So if you start from 1 pyruvic acid, you get 1 NADH2 and 12 ATPs. 1 NADH2 is going to give you 3. So you will totally get 15 ATPs. How many ATPs will you get? If you start from pyruvic acid, which goes complete oxidation. So your answer of 12 ATP is not right. Did you understand it? Why our answer for this is 15 ATPs. Is it clear now? Anyone who has doubt in this first question? Hiren, Kalai, both of you wrote 12 ATP. Even Rashmi said. Okay, see, our question is 1 molecule of pyruvic acid when it is completely broken down aerobically, then how many ATPs will we get? See, 1 pyruvic acid you are starting with. 1 pyruvic acid, if it has to enter Krebs cycle, then it has to undergo that link reaction to form acetyl coenzyme A. And during this link reaction, 1 NADH2 will be produced, right? And if 1 NADH2 is produced, that means it is going to give you 3 ATPs later on. And this acetyl coenzyme A, when enters Krebs cycle, then it gives us 12 ATPs. So 3 from NADH2 and 12 from here. That's how we are going to get 15 ATPs. Is it clear now? Kalai and Rashmi, both of you wanted to know this answer. So is it clear now, the first question? How we got the answer as 15 ATP? Very good. What about you, Kalai? Rashmi says she understood. Okay, shall we come to second question? If you have 1 molecule of PGAl, how many ATPs will you get on its complete oxidation? Where is PGAl? Glucose, glucose 6-phosphate, fructose 6-phosphate. We are talking of glycolysis. Then fructose 1,6-biphosphate. And then this dissociates into 1 molecule of 3-PGAl. Then what happens is, from this PGAl, we will get 1 pyruvic acid. And in this process, we will get 2 ATPs and 1 NADH2. Right? So don't forget to count this. Then next step, are you counting it? This is 2 and 1, that is 3, 5 ATPs from here. Then this enters into link reaction. This link reaction gives us NADH2, that means 3 ATPs here. And then Krebs cycle, that is 12. So 5 plus 3 plus 12. What is the number? How many ATPs will we get? If you are starting from one molecule of 3-PGAL, that means you've started from here. When fructose 1,6 biphosphate breaks into two compounds, you're talking about one. We got one NADH2 and two ATPs, then a link reaction, and then Krebs cycle. So you'll have to add 5 from here, 3 from here, and 12 from here. Got it? So here we will get 20 ATPs. Is this clear now? Alright, let us come to the third question. Where are the enzymes for TCA located? This is our third question, and herein answered enzymes for TCA are present in mitochondrial matrix. So this answer is right. TCA is Krebs cycle, so it is in matrix of mitochondria. So this answer is correct. What is the number of ATP obtained from one NADH2 and one FADH2? This is very simple. One NADH2 is going to give you 3. Hiren has answered that question, and one FADH2 gives you 2 ATPs. Right? So here we were not asked to draw anything, any pathway, any cycle, nothing. The questions were asked from the pathway. And that is why we should be knowing this pathway very, very clearly. Okay, now let us come to the next thing which we want to start. See, where are we right now? We are on the fourth step. The first step was glycolysis. The second step was link reaction. Third step was Krebs cycle. And the fourth is electron transport chain. And this is what we will be talking about in detail today. And the last is oxidative phosphorylation. So we plan to discuss these two steps, and this will complete our aerobic respiration. Okay. I'm going to tell you brief idea of electron transport chain, and then we will see the actual electron transport chain which is given in our NCERT. So if, say this is the mitochondrial membrane, I am drawing an outer mitochondrial membrane. This is the outer membrane, and then let us say this is the inner membrane, and I am showing this crista. We will draw the complete electron transport chain one more time, detailed chain. This is just to understand the concept. Okay, this membrane is the inner membrane. This membrane is the outer membrane. All right, this space is called the outer compartment, and the inner side is all matrix. Now when we are talking of the membrane, in the membrane are present some integral proteins. Integral proteins, that means they are embedded inside the membrane. So this is one protein, this is another one, this is another one. There are many such proteins which are in the membrane. Okay, and they will make the electron transport chain. Through them the electron will get transported. That is why we are calling them electron transport chain. Now where is this electron going to come from? The electron comes from NADH2. Where did we get NADH2 from? We got NADH2s from various places. Link reaction also gave us NADH2, Krebs cycle also gave us NADH2, and this is the matrix. So we are talking about the NADH2s which are in the matrix. So this NADH2, it dissociates into NAD, into proton, and into a pair of electrons. And these electrons, they pass through these proteins. So when the electron pass through these proteins, this electron is moving from higher energy level to lower energy level. So when it goes from higher energy level to lower energy level, it will release that extra energy. See, suppose we are talking about these steps. These are the steps, and you have a ball which you want should come down to the lower point. Here it is at a higher level and you want it to come at the lower level. Suppose you throw this ball directly here, the complete energy will be released in one step. That energy released in one step may not be used inside a cell, because cell can use energy slowly. So you want that energy should be released slowly. So what we do is, we let the ball hop and jump, 2 and 3 and then down. So at 1, 2 and 3 place, some energy is released. If inside a cell the same things happen, if slowly energy is released, then that energy can be used. So when this pair of electron passes through the electron transport chain, it releases energy at 3 steps. So 1 step energy released, 2nd step energy released, and 3rd step energy is released. Now what is done with this released energy? This released energy is used to pump. And I'm using the word pump, that means we are talking about active process. To pump a pair of protons from the matrix into the outer compartment. Are you getting it? Is this much part clear? Because this is just the basic. If this basic part is clear, then we will come to the actual electron transport chain, which is going to look little complicated. Are you getting this point which I'm trying to tell you? That NADH2 dissociates. A pair of protons is released, NAD is released, and electron pair is released. Now this electron pair is at a higher energy level. When it goes through all those proteins, it releases its energy at 3 places. And at every place, whenever energy is released, using that energy, a pair of proton will be pumped into the outer compartment. Energy released, pair of proton pumped in. Energy released, pair of proton pumped in. At 3 places energy will be released, and at every place a pair of electron is, the energy which is released is enough to pump 2 electrons and a pair of protons. Sorry, this is also a pair of proton and this is a pair of electron. So 3 places energy is released and 3 protons are sent from the matrix into the outer compartment. And this electron which is coming down the energy level is finally accepted by oxygen. So finally it will be taken by oxygen. We write it as half O2 by 1 atom of oxygen. So this oxygen will get the electron, it will get the proton pair, and this is how we get water. Okay, so in respiration, is water produced? What is the reaction of respiration? C6H12O6, glucose broken down in presence of oxygen. Aerobic respiration is what we are talking about. And where is this glucose broken down? Glycolysis. And in this, what do we get? Carbon dioxide is given out, plus water is given out, plus energy is released, right? So where is this water coming from? This is how this water is formed. Clear? So what has happened is, by using this NADH2, which is formed in various reactions, a pair of electron passes through a chain of proteins, and during its transit through that chain, at 3 places energy is released. And whenever energy is released, a pair of protons from the matrix will be pumped into the outer compartment. So now what is happening is, in the outer compartment there is higher and higher concentration of protons. So now there is a proton imbalance. In the outer compartment more protons, in the matrix less proton. This is concentration gradient. See, higher energy, lower energy, this is energy gradient. Higher concentration, lower concentration, this is a concentration gradient. Gradient is the difference between the highest and the lowest levels. Now, this part, if you talk of, this is electron transport chain. And if you talk about the protons coming back into the matrix, that is through F0, F1 particles, then that step is oxidative phosphorylation. Okay, so I have given you some basic thing of what happens when one NADH2 dissociates. And when one pair of proton comes back into the matrix, it gives us 1 ATP. So here one proton comes back, it releases that energy, and that energy is used to add phosphate to ADP, and we would get ATP. How many proton pairs have been sent out from one NADH2? 3. One goes back, 1 ATP, second goes back, second ATP, third goes back, third ATP. That is why we say that one NADH2 is going to give us 3 ATPs. Is this part clear? Why one NADH2 is worth 3 ATPs? Because when one NADH2 dissociates, the electron pair comes down the gradient through electron transport chain, and at 3 places protons are pumped, proton pairs are pumped from matrix to outer compartment. And when these protons go back into the matrix, every time 1 ATP is generated. So one proton coming back, 1 ATP, second ATP, third, 3 ATPs. What will happen if FADH2 dissociates? Instead of NADH2, if FADH2 dissociates, then what will happen? Sabarish Kumar is saying, ma'am, please talk in English in Unacademy special class. See, Sabarish, for the students who are exclusively interested in English, pure English lectures, for them this is the channel where we are right now. Now on special, there are some students who are like little more comfortable with Hindi-English mix. But terminology is English, so it is 80 to 90% English, but you know, to make them understand or to make them little more comfortable, the filling sentences are in Hindi. So I don't think there should be any problem. Rahul says, ma'am, you missed the beginning. Okay, Sheikh Bilal is sending some emojis. Okay, so before we actually take up the electron transport chain, I'm going to just show you the picture for a minute and then we'll come back to this. This is the actual electron transport chain. Now, if I would have shown you this directly, probably some of you would have, gotten scared. But to understand, I made a very simple diagram. This is the outer mitochondrial membrane, this one, outer, and then this is the inner mitochondrial membrane. Mitochondrial membrane is like plasma membrane, and there are some proteins which are embedded in the phospholipid bilayer. We call them integral proteins or intrinsic proteins, and these proteins they actually make up the electron transport chain. It is a chain of proteins through which the electron will get transported. Now, from where is the electron going to come? One such molecule is NADH2. When NADH2 dissociates, a pair of electrons is released. And this electron is at a higher energy level. From higher level, it comes to lower energy level. And when it moves from higher to lower, it passes through those electron transport chain. And at 3 places, it releases energy. One place energy released, a pair of proton from the matrix is pumped into the outer compartment. Second time energy released, a pair of proton pumped in. Third time energy released, a pair of proton pumped in. The proton pairs can come only through F0, F1 particle. So when one proton comes out into the matrix, one ATP is synthesized. So there are 3 places, or rather 3 proton pairs which are coming in. So 3 will go out, 3 pairs, and that is why 3 ATP. Suppose one FADH2 dissociates, again there will be FAD, a pair of protons and a pair of electrons. But when the pair of electron passes through the electron transport chain, it releases energy only at 2 times. So energy released, energy released. And whenever energy is released, that proton pair is pumped in. Second time energy released, proton pair is pumped in. So how many pairs have been pumped in? 2. That means 2 pairs will go out. So when first pair goes out, 1 ATP, second pair goes out, 2 ATP. That is why FADH2 gives us 2 ATP. Ma'am, hydrogen ions will be there in case of FADH2 also? Yes. FAD, a pair of protons and a pair of electrons. Clear? If this part is clear, then only we can go to the next diagram. Any confusion in this part? Because this is just the basic. Clear? Sheikh Bilal says clear, Rashmi is okay, Kalai says clear. Okay, so that means we have understood the basic. Now, let us come to what is the difference between NADH2 electron and FADH2 electron? See, there is no difference between the electron. The difference is in their energy level. The electrons which are released from NADH2, they have more energy, because rather they are released at a higher energy level. So when they come to the normal energy level, the energy is released at 3 steps. Whereas in case of FADH2, they are also at higher level, but they release the energy only at 2 steps. And that makes the whole difference. Okay, now let us see the electron transport chain which is the actual electron transport chain, and we need to know it completely. Now this labels, let us check this label. This is the inner mitochondrial membrane. This is the membrane space, and somewhere here is the outer membrane. The outer membrane is not shown in this diagram, only inner membrane is shown. So where is this electron transport takes place? In inner mitochondrial space. Sorry, inner mitochondrial membrane. Alright, now here NADH is dissociating. We can write it as NADH plus, or we can also write it as NADH2, both are correct. And see, this electron pair is released. This whole thing which is given here in this box, this whole thing is called a complex, and we name it as complex 1. So this complex 1 is mainly NADH dehydrogenase. It is going to remove hydrogen from this NADH2 or NADH plus. So this complex 1 is, its job is, it is acting as NADH2 reductase or dehydrogenase. NADH2 dehydrogenase. What is it made up of? This complex is made up of two proteins, FMN and FES. What is FMN? It has two. It has FMN, that is flavin mononucleotide, and FES, this is iron sulfur containing protein. So these two make complex 1. And what is the job of complex 1? To remove the protons from NADH2. Clear? Now this electron pair is going to come down the gradient. Here you see UQ written. Okay, I'm writing here. UQ is ubiquinone. And then there is UQH2. This is reduced ubiquinone. Now, what is reduction? Do you know reduction is? Reduction is gain of electrons. So UQ gains electrons and then it changes into UQH2. Alright, now it comes to complex 3. This is complex 3, or let us write it here, complex 3. What is the job of complex 3 and what is it mainly made up of? It is mainly made up of cytochrome complex. See, in this complex you have cytochrome B, then there is iron sulfur containing protein, then there is cytochrome C1, and that is why we call it cytochrome complex. So cytochrome complex is the complex which is made up of various types of cytochromes. Cytochrome is an iron copper containing protein, and depending upon more iron or more copper we have different types of cytochromes. Okay, now next is complex 2 and it is succinic dehydrogenase. Succinate dehydrogenase. That means these are the enzymes or molecules which are also acting as electron transporters. So here is this succinate changing into fumarate also. this succinate fumarate? See, I know some simple formula. So this is simple formula. So here there is this enzyme, and this enzyme is also acting as the electron transporter. So this protein, complex 2, is doing two jobs. It is converting succinate into fumarate, and at the same time it is also helping in transport of that electron. So now the electron is moving through here, and then it finally comes to this oxygen. See, finally electron is accepted by oxygen. So here last statement is final electron acceptor is oxygen, and that is how you get water. Now this is fourth complex, that is complex 4. Complex 4 is cytochrome oxidase. Now all these names, they may sound little confusing, but see what is happening here. When the electrons are lost, they are coming into, or they are releasing the energy. At complex 1, that energy is used to pump proton. This is place number 1. Second place, protons are pumped, that is complex number 3, and third place where the protons are pumped is next place, that is complex number 4. Now in the previous diagram which I showed you, we said NADH2 releases a pair of electron, and this pair sends one pair of proton, second pair of proton, and third pair of proton. When one FADH2 dissociates, it releases one pair of proton and second pair of proton. So if you put this here, that means if one FADH2 is also dissociating, NADH2 and FADH2 together, so a pair of proton from NADH2 and a pair of proton from FADH2. So this will make it 4H plus. One pair of proton from NADH2 and one pair of proton from FADH2. This will make 4, and there is only one pair from NADH2. Okay. Now let us come to this. So here, how many protons are we seeing? 4. 2 through NADH2 and 2 through FADH2. So here they have written only NADH2, but we have to understand that there is FADH2 also which is dissociating. Again we have 4 here, so 2 protons, that is one pair from NADH2 and one pair from FADH2. Here also same, from NADH2 and one pair from FADH2, and the last one is only from NADH2. That is how here the number, this part is actually oxidative phosphorylation. So in electron transport chain, what happens basically, there are complexes. Complexes are clusters of proteins. And when electron passes through these complexes, at 3 places energy is released if the electron is coming from NADH2, and energy is released at 2 places if the electron is coming from FADH2. Which are these complexes where energy is released? Complex 1, complex 3 and complex 4. These are the ones, and complex 2 is mainly acting as an enzyme. So there are 1, 3 and 4 at which energy is released, and wherever energy is released, a proton will be pumped from the matrix into the outer compartment. Is this electron transport chain clear? Cy whenever we write, it is cytochrome. Fes when we write, it is iron sulfur containing protein. Fmn is Fleming mononucleotide. Okay? Yes, ma'am, very good. So, this is why we start with a very simple explanation, because if we see this complex thing at the first place, then it is slightly confusing. So what we do is we study in a simple manner and then we come to this complex. It is not that complex, but yes, when you're doing it for the first time, it looks complex. Now, I'm going to draw again a very simple diagram for you. This is the outer membrane of mitochondria. This is the inner membrane of mitochondria, and this is crista. The folds in mitochondria, inner mitochondrial membrane are called crista. This, label this again, outer membrane, inner membrane, and this is the outer compartment. And what is here is all matrix. Electron transport chain. Let us draw that electron transport chain in a very simple manner again. Say here are these proteins through which the electrons are going to travel. So here NADH2 dissociates, we get NAD, we get a pair of protons and one pair of electrons. Okay? Now this electron goes down the gradient. It passes from here, all these electron acceptors, and finally this electron pair has gone to O2. This O2 also takes a pair of protons and now it changes into water. This is the metabolic water which is formed. We call it metabolic water. When this electron was passing through the electron transport chain, at 3 places energy was released, 1, 2 and 3, and by using this energy, a pair of proton from the matrix was pumped into the outer compartment. A pair of proton, by using this energy, was pumped in. Again, 3rd place, it was pumped in. So now what has happened inside the outer compartment? Here, the concentration of protons has increased as compared to the matrix. Now here there are protons accumulating. These protons cannot go back on their own through the membrane. They can go back through a very special protein which is called F0F1 particle. This is embedded in the membrane. This is called F0 particle or base. The middle one is called F5, F6 particle or stalk. And this is called F1 particle or head. And you know this head, it acts as an enzyme, and that is ATPase. So what do you think ATPase will do? ATPase will help in formation of ATP. Now, this proton comes back from the outer compartment through this F0 stalk, and here in F1 it releases its energy. And then without or with minimum energy, the proton comes back into the matrix. Now when it releases all that extra energy, so that energy is used to add inorganic phosphate to ADP, and what will we get? We get ATP. So one proton goes back, we get one ATP. Second pair goes back, we get second ATP. Third pair goes back, we get 3 ATP. So from one NADH2, 3 pairs of protons were sent from matrix to outer compartment. When these 3 return, we get 3 ATPs, and that is why we say one NADH2 is worth 3 ATPs. From where is this O2 coming? O2 is always there in cytoplasm, little O2 is dissolved. This is the same oxygen which we are breathing in. And because oxygen is involved, this is a step of aerobic respiration. So is oxidative? Oxidative because oxygen is a part of it, electron, and phosphorylation, that is addition of phosphate, and that is what is the name of this step, oxidative phosphorylation. Rashmi, you understood it, very good. Kalai also understood, Hiren also understood. So oxidative phosphorylation is the place where we actually get ATP. And where is the ATP synthesized? These particles, all these 3 together, this whole thing is known as oxisome or F0, F1 particles. Ma'am, Rahul says we are reading about plants, right? Because chapter named is respiration. That's right. Oh, so you mean to say plants don't take oxygen? Whenever respiration is there, plants also take oxygen. Respiration is always taking in oxygen and giving out carbon dioxide. When they have to perform photosynthesis, that is when they take carbon dioxide and give out oxygen. Right? So it is the same oxygen which they are taking in during respiration. Rajni Kant, good evening, hello. Proteins of electron transport chain are a bit confusing. Yes, I know, because when you do it for the first time, it sounds confusing. But here, if you read this 2-3 times, your main focus should be on the complexes. It is first complex, then third, then second, and then fourth. And the second complex main function is acting as an enzyme. But first, third, and fourth are the ones where energy is released from that electron. The first one has FMN, FES. One is FMN, flavin mononucleotide. Second is iron sulfur containing protein. The third complex is all cytochrome and in between one iron sulfur containing. And the fourth complex is again cytochrome. Okay, so if you do it 2-3 times, you will remember this. Rashmi says, ma'am, in respiration they need oxygen. Yes, they do. Every cell, unless and until it is an anaerobic organism or a bacterium, they need oxygen for survival. And obviously oxygen is required for aerobic breakdown, because aerobic breakdown is going to give us more energy. In between complex 1 and 2, complex 3 is present. Yes. Complex 2 is an enzyme. And 1, 3 and 4 are the places where energy is released. Okay, so questions asked are, which is the final electron acceptor? It is oxygen. How many pairs of protons are pumped from matrix to outer compartment when NADH2 dissociates? The answer is 3 pairs. How many pairs are pumped when FADH2 dissociates? Answer is 2 pairs. How many ATP is synthesized from NADH2? 3. From FADH2? 2. Which complexes are those where the energy is released? It is 1, 3 and 4. What is the main role of complex 2? It is helping in conversion of succinate into fumarate. Okay, so if this is clear, then let us have a quick recap of what has happened so far. See, up till now what we have studied is, in glycolysis, we got 2 molecules of pyruvic acid, we got 2 NADH2 and 2 ATPs. After that thing was settled, in investment phase we took 2 ATP and then we gave it back. So finally we got 2 left. Then link reactions. Link reaction takes place 2 times because every pyruvic acid changes into acetaldehyde. In this, 2 pyruvic acids are converted into 2 acetaldehydes. Acetyl coenzyme A. Acetyl coenzyme A. And in this, 2 carbon dioxides are given out. But in this process, we also got 2 NADH2. Then the next step is Krebs cycle. In Krebs cycle, from 1 glucose there will be 2 Krebs cycles, and 1 Krebs cycle gives us 3 NADH2, 1 FADH2 and 1 ATP. And this has to be doubled because 1 acetyl CoA will go through 1 cycle, second molecule will give us second. So this is going to give us total 24 ATPs, right? So this ultimately is going to help in that balance sheet. Fourth step was electron transport chain. In electron transport chain we are not getting anything. Here NADH2 and FADH2 dissociate to release electrons and protons, and it creates a gradient, a proton gradient. And this proton gradient is useful in ATP production. So here proton gradient results in ATP synthesis, and that is how we get ATP. So see, first 3 steps, glycolysis, link and Krebs, they have given us something, either ATP, which is like cash, or check in the form of NADH2 and FADH2. If you remember, we were talking about that when we go to the bank, there is a counter where we deposit the check, and then there is a counter from where we collect the cash. So ETC, electron transport chain, is a counter where you put in the checks in the form of NADH2 or FADH2. And oxidative phosphorylation, that is oxysomes, is the counter from where you get ATP or the energy currency of the cell. So we say mitochondria is the powerhouse of the cell because ATP is produced here. But now we know precise location where ATP is produced. And this ATP is always generated because of proton gradient. The same thing happened in chemiosmosis. in chloroplast, and the same thing is happening in mitochondria. In chloroplast, the particles were called F0, F1 particles, and here the particles are called F0, F1 particles or oxysomes. What is the difference? In this case, the particle has F5, F6, whereas in chloroplast there is no F5, F6. It is only F0 and F1, and that is why in chloroplast we call it F0, F1. Here we call it F0, F1 or oxysomes or elementary particles. So when you use the term oxysome, three parts must be there: base, the stalk and the head, and the head acts as ATPase, the enzyme which helps in phosphorylation or ATP synthesis. It is the enzyme that helps in ATP synthesis. And how is ATP synthesized? By phosphorylation, addition of phosphate. So is ETC clear? Is oxidative phosphorylation clear? If these two steps are clear, then we are done with cellular respiration part. Now we are left with only three simple things. We have to have an energy balance sheet. We will talk about a common pathway. Suppose instead of glucose, there is something else, say protein is there or fat is there. Do we still get energy? Answer is yes, and there is a common pathway. We will talk about that tomorrow. Rashmi says everything is clear. What about others? Did you understand these two important, very, very important steps? ETC and oxidative phosphorylation. I guess it is clear. You do one thing, you revise these two things from NCERT, read the book, read what is given there. Here we have used the language which is easier for us to understand, but in a book they write in a different manner. It is called bookish language. But if you understood everything, then you will be able to understand that bookish language also. So Rashmi, Hiren, Rahul, everyone says that it is all clear. Kalai also says clear. So, okay, so I'm happy that you have understood all the steps. Now in the next class, that is tomorrow, what we will do is, we will finish this chapter, we'll wind it up. And after that, we will be having a doubt solving session and a quiz session. So, why you don't speak in Hindi? See, I know Hindi, I can speak in Hindi, but this channel is Let's Crack NEET UG English. So it's a pure English channel. On other channels, on Unacademy Learning app, I do speak in Hindi. मुझे हिंदी बोलना आता है, and I can talk to you in Hindi. But this channel is specifically for those students who want pure English. ठीक है? So हिंदी में बात करना है, so we'll have to come to some other channel. And अगर biology नहीं पढ़ना है, तो we can talk in Hindi. But if it is biology, then on this channel it's going to be English only. Okay, next chapter. Next chapter is going to be transport in plants. We will start with transport in plants. And I have told you that I'm going to have a special video which is like telling you the strategies to score 360 on 360 in biology. And that is going to be day after tomorrow. I think, no, it is on 31st, I guess. It is Saturday, okay, I don't remember the date, but it is Saturday, and the timing is 2 to 3, and that is we will not be talking about the study part. It is going to be just telling you how you can score 360 on 360. So do join in. Ma'am, is NCERT not in? Oh yes, it is, because everything is NCERT based. It is not enough for 340, I would say it is enough for 360. Only thing is the way you study NCERT is the most important thing. And this is what I'm going to be telling you on Saturday. Alright, so I'll see you tomorrow and we'll finish this chapter. And don't forget, Saturday 2 to 3 to talk about the strategies. Alright, so till tomorrow, take care. Bye.